to continue on with our acquired immunity talk, and we finished talking about cell-mediated immunity. We talked about the activation of the T cells, the cytotoxic T cells, and the helper T cells. Now we're going to talk about antibody-mediated immunity. So let's go on. We've got 4 steps in antibody-mediated immunity. Step 1 is called B cell sensitization. Step 2 is called B cell activation. In step 3, the activated B cells will divide, and then they will form what happens in step 4, which is antigen/antibody complexes that result in many, many effects. Let's start with step 1, our B cell sensitization. This is the preparatory phase, preparation for activation. And what happens here is the in sensitization, the antibody molecules that are specific to the antigen are already present on the B cell membrane. So let's look at some of these subpoints. Antibody molecules already present on the B cell membrane. They're already present. Oops. The antibody molecules that are specific to the antigen are already present on the B cell membrane. So we need to know that. The antibody molecules. So we need to talk about antibodies here in a little bit. We need to note that the antigen binds to the antibody on the cell membrane. I'm abbreviating, but it really kind of saves time. The antigen enters the cell through endocytosis, and then the antigen will reappear on the cell surface bound to class II MHC protein. Okay, so that is the sensitization stage. That's kind of getting sensitive, being sensitized to the antigen. So the antibody molecules are already on the B cell. The antigen in question, the bad guy, binds to the antibody. The antigen will then enter the cell through endocytosis. The cell will take in the antigen bound to the antibody. And then the antigen will then reappear on the cell surface bound to a class II MHC protein. Let's see if I have a picture. Here, let's just look at sensitization. There, if you look a little more closely, you might have to get your book because this is kind of small, but this shows that we have antigens. The little red things are the antigens floating around. The antibodies look like little Y-shaped things on the surface. And then we also are showing some class II MHC proteins on the surface of the cell. And the antigens will bind to the antibody on the B cell, the inactive B cell, because there's a special receptor. They act like they fit specifically. The antibody has a special binding site and it will bind perfectly. Once that binding occurs, then it's taken into the cell, and then the antigen is stuck out or presented on the outside there on the MHC2 protein. So that is sensitization. All right, let's go to step 2, which is B cell activation. Now the B cell is going to encounter or come in contact with an activated helper T cell. We remember that the helper T's got, these are also called the CD4 cells, so I'm going to put CD4 cell. We remember that from our cell-mediated immunity. The helper T, or the CD4, is going to bind to the MHC complex. It will secrete cytokines that do a bunch of things. What do the cytokines do? Well, in our in our lecture outline, we say that these cytokines are going to promote, B cell activation. They're going to stimulate B cell division. They're going to accelerate plasma cell formation. Plasma cells are the cells that produce antibodies, and they're going to enhance antibody production. Okay, now what do these activated B cells divide into? What do they divide into? They divide into memory B cells, and they will divide to become plasma cells, which will then secrete large quantities of antibody. Okay. So this is the picture again. We talked about the sensitization. Now we've got a sensitized B cell, and he's going to run into an activated helper T cell. This activated helper T, now we're going to blow up this little interaction between the two. Here's the B cell. There's his there's his little B cell receptor, and he's holding out the antigen, and there is the T cell, and the T cell has the class 2 MHC receptor, or the class 2 MHC protein, and this then we have, so anyway, this is where, and both of these are binding to the antigen here. So this is showing that the T cell is activating the B cell. The helper T is activating the B cell. Okay. And once this guy the B cell is activated, it also is the T cell is doing some very important things. The T cell is sending out cytokines that are going to encourage so these helper Ts are great big helpers. They will send out cytokines that are going to stimulate the plasma cells. Well, first of all, they're going to stimulate the helper T cells to become I'm sorry, and I can't rewind this guy. So as I said, the T cell, help me, just hang in here. These helper T cells are going to cause more helper T cells to be produced. So they're going to increase the helper Ts. They're going to help activate the B cells. They're going to stimulate these activated B cells to produce or to become plasma cells, and then they're also going to stimulate the plasma cells to produce lots of antibodies. So there's just lots of stimulation here. The T cell is going to stimulate, he's going to activate the B cell, he's going to also stimulate other helper Ts to do the same thing, to help activate the B cells and to help make them turn into plasma cells and produce more antibodies. Okay, so this just breaks down our sensitization phase, where the antigen, at step 1, antigen is going to bind to the antibody, which gets taken in, and then it will present the antigen out here on the class 2 MHC. This thing is, this is showing you again the B cell. This was sensitized. The B cell is sensitized. There's the antigen. Here's the helper T cell, and it's going to secrete cytokines that are going to help activate. Once this binding occurs, it's going to send cytokines that are going to activate this B cell. Okay, and then we've got all these cytokines that are doing things that we have discussed already. They're going to be triggering, continuing to activate B cells. They're going to stimulate the B cells to become plasma cells and encourage them to make lots of antibody. Okay, and also in the process, we're going to have memory B cells that are going to be waiting around just in case there's exposure to the same antigen down the road. So here's a question for you. B cell sensitization occurs when the antigen binds directly to the class 2 MHC proteins, the antigen binds to the antibody that's present on the B cell, the antigen enters the cell by endocytosis and is attached to the class 2 MHC protein and then is inserted into the cell membrane, or is it B and C, or is it none of the above? So if you go back and look, you should be able to see where that is, what the correct answer is. And you can do this one as well, and we'll let you look at those. So let's just talk a little bit about antibodies. Antibodies are molecules, they're proteins. These are proteins, so don't forget that they're proteins. And they consist of one pair, meaning 2, heavy chains and one pair of light chains. Each chain contains both constant segments and variable segments, and we'll tell you what those mean in just a minute. There are 5 types of B cell antibody constant segments, and we call these IgG, IgE, IgD, IgM, and IgA. Now the variable segments are called the antigen binding sites. This is where the antigen actually binds. And because antigens are also different, the part of the antibody that's variable or different than the others is the part where the antigen binds. It makes total sense because the antigens are all unique. But the constant part is the part that's the same on all of these different kinds of antibodies. And we have 5 main kinds of antibodies. And we need to remember that disulfide bonds connect the light and heavy chains. They're the type of bond that involves sulfur. Okay, make that stop. Okay. This is an image of an antibody. They're kind of shaped like Ys. All right, we've got the heavy chains are these. So these are the heavy chains right here. This is one heavy chain. There's the other heavy chain. Okay, and then the light chains are these little short pieces that stick off the arms, so to speak. So this is a light chain, this is a light chain. Now the variable part of the molecule, the variable segment, is the part that's got hash marks. And these are this end. This is where the antigen will bind. The antigen binds right here to the variable segment. The antigen will bind right here to the variable segment of the antibody. Okay, so, and then we have this little area here, kind of like, if you think of this as little arms, and that's its little body, I think of this kind of as the little armpit, and this is where the complement binding site is. Right there, right under the little armpit here, the complement binding site. And then these are the sites where they bind to macrophages. I'm not worried about that as much, but just kind of keep that in mind. Okay, these are your classes of antibodies. In your lecture notes, you've got where these are all found, what their role is, and these are things that you'll want to review and go through. So I'm going to give you just a little bit of time to go through these. These are things that I have lectured, or, well, actually, you know what? I'm going to do this right now. I'm going to take you through these. IgG. I'm going to type this in some small font as we go, and we'll just talk about them right now. IgG is the most diverse group. It's the most numerous group. So in other words, the most antibodies are in the IgG group. They cross the placenta, and they're then responsible for passive immunity to the fetus. Okay, that's IgG. Okay, IgE is attached to basophils and mast cells. Basophils are circulate in the blood, and when basophils are found in tissues, they're called mast cells, and they release histamine. Remember, histamine is a vasoactive chemical that's involved in inflammation, causes the cardinal, those 4 signs of inflammation that we've talked about. So IgE is important in the inflammatory response. Okay. IgM is the first class produced or secreted. The levels go down when or as IgG increases. So it's the first class that is secreted once you have an infection. Oops, that was supposed to be IgM. Sorry. So let's just do this. Sorry about that. We'll just make this IgM. IgM is the first class secreted, and the levels go down as IgG increases. And sorry about that. Let's talk about IgD. IgD is interesting because it's found on the surface of B cells. It binds antibody, or excuse me, it binds antigen. It binds antigen. So IgD is an antibody that is found on the surface of your B cells. So it binds antigen. And then and actually if we go back and we remember the process, let's go back and look at the process of sensitization. These antibodies that are on the surface of the inactive B cell, which antibody do you think this is? That's on the surface of that B cell, the B lymphocyte. It would be IgD, right? IgD. It's found on the surface of B cells. It will bind antigen. So it's important in antigen sensitization. And then IgA is found in secretions, such as saliva, tears, mucus, semen, and what they do is they will attack pathogens in those secretions. All right, so they're very important in, what's another secretion in mothers? Breast milk. Okay, so IgA is in secretions. So the antibody type that's attached to basophils and mast cells, okay, you need to look that up and do it. Okay, so now let's just talk about some things related to antigens, and actually I think we're going to stop. We won't talk about things related to antigens. We'll take this up in our next video. Thanks.